Greetings, ladies and gentlemen. Once again, I want to welcome you to my YouTube channel. In today's class, we are going to be talking about the animal kingdom holistically, and we are going to be picking some of the phyla one after the other. So, the animal kingdom. Let's x-ray the animal kingdom. When it comes to the animal kingdom, we have about 10 different phyla that are found in the animal kingdom. And when I was a student, there is this acronym that I derived, which can easily be used to remember all the phyla in animal kingdom. And I want to write out this acronym. I wrote P squared CPN, then amic. P squared CPN amic. P squared CPN amic. So with this, I can tell you the different phyla we have in the animal kingdom. So let's go. The first one, so let me write this upwards. P squared CPN amic. So I have the first P as protozoa. I have the second P as porifera. I have C as cnidaria. I have P as platyhelminthes. We have N, C, P, there's a mistake somewhere. No, it's correct now. So this is the first P protozoa. Okay, yes. Yeah, that's correct. This is P squared, then CPN, N, Nematoda, then A is anellida, then M is mollusca, that's where you see the snails, then A is Arthropoda, then E is echinodermata, then C is chordata. So protozoa, porifera, cnidaria. This is also called the colentrata. The cnidaria colentrata, platyhelminthes, Nematoda, anellida, mollusca, Arthropoda, echinodermata, then chordata. So these are the 10 phyla we have in the animal kingdom. Okay, having known this now, let me talk about this chordata. As a matter of fact, the chordata is the phylum that contains both the vertebrates and the invertebrates. So let me talk about the chordata. This chordata is made up of 4 different subphyla. The first one is Chordata. This is where you see the 6 words, or the tourniquets. They'll have the second one. The second one is Hemichordata. We'll have the ascon worm. Then we'll have the third one, which is Cephalocodata. Cephalocodata. This is I have the amphiosus, amphiosus. Then I have the fourth one, which is vertebrata. And of course, you know vertebrata is made up of the fishes, the pieces, the amphibians, the reptiles, the eels, and the mammals. So can you see that all the animals you can think of that have been discovered are found within this whole animal kingdom, from protozoa down to the last one, which is chordata. And like I said, this chordata is a phylum that links the invertebrates and the vertebrates. So from protozoa down to cephalochordata are called invertebrates. Then vertebrata, containing the five classes, are called the vertebrates. So that is how to differentiate between vertebrates and invertebrates. But then our interest for today's class is to study the invertebrates. The invertebrates. And in the invertebrates is just one of the phyla I want to look at. So I said we have P, C, P, N, and Mike. From this to this are known the acylommates. Are known as acylommates. Then this is pseudocoelomates. Then this is coelomates. What do you mean by coelom? What do you mean by coelomates? When we say that an animal is coelomate, that means that that animal contains a body cavity, and that body cavity is called coelom, which is lined by a tissue, the mesoderm. And of course, for most of these animals, or this phyla that contains animals that are a bit more complex than the non-coelomates, they have they are supposed to have this coelom, this body cavity that helps to protect their organ. It helps to protect their organ, and of course to also cushion the effect of friction. And for some of them, the, uh, the body cavity or the coelom serves as a kind of skeleton that also aids their movement. That's what people call hydrostatic skeleton. But for these non-coelomates, this group of animals are not very much complex, and of course they also possess less complex structures or organs, and as a result of that do not need a coelom for anything. So that is why there is no coelom in this group from protozoa down to platyhelminthes. Members of nematode have coelom, but the coelom is not a true one, or they have, let me say, they have false coelom, just like you have false feet in amoeba, and that's what they call the pseudopodia. So they have false coelom, and that's what they call the pseudocoelomates. And nematoda is the only animal, or the only group of the only phylum that is categorized as the pseudocoelomates. So having given this introduction, let's now look at protozoa. Protozoa is the first animal. Proto, first. The zoa means animal. The first animal. And as you can see in the outline I gave, protozoa is the first one. So when we talk about protozoa, you discover that we want to look at the characteristics. Number one is that they are unicellular organisms. They are unicellular. So the first one is that they are unicellular. Unicellular. What do we mean by unicellular? When you say that an organism is unicellular, That means that it contains a single cell. All the organelles are retained are contained within a single cell. So that is what we mean by the word unicellular. And apart from the fact that protozoa are unicellular, they are also microscopic, microscopic animals. So protozoa are unicellular, but they are also microscopic. And what does it mean to be microscopic? It means that a microscope is required to be able to see them clearly. That is number two characteristics. Then number three is the fact that protozoa have a different method of movement. They can move through the use of cilia. They can move through the use of pseudopodia. They can move through the use of flagella. They can move through the use of undulating ridges. Undulating ridges. So cilia, members of protozoa that move through the use of cilia. Of course, paramecium for free-living protozoa, the Balatinum coli for parasitic protozoa. Then pseudopodia for free-living protozoa, amoeba proteus for parasitic protozoa, and amoeba isolatica. For flagella, of course, we have the trichomonas as the parasitic protozoa that has flagella. Then chlamydomonas, volvus, these are also protozoa, but they are free living, they are not parasitic. The undulating ridges, you see this in members of plasmodium. That's a kind of wave action that is created within the cytoplasm, and it also helps to extend them or to move them from one position to the other. So these are the locomotor structures that are found within protozoa. Then it is also important you know that some members of protozoa, like I just explained, are free living, while some are parasitic. When you say that an organism is free living, that means that that organism does not live in or on the body of an organism, deriving nourishment at the expense of the host, and this defines parasitism. So some members of protozoa live on their own within the environment, like I remember amoeba proteus. Amoeba proteus, then paramecium, chordatum. So these are, and others, these are lists of protozoa parasites that are free living. They are not parasitic. Then the body of protozoa is naked, being made of only cell membrane. Their body is naked, being made up of only cell membrane. And of course, gaseous exchange occurs within this cell membrane. So the fact that they have cell membrane which covers their body is the reason it is said that their body is naked. However, there is a member of protozoa that is called actinophrene that has a shell that is being covered by a shell. Actinophrene. So this example is an example of protozoa that is covered by shell. Then what again means of reproduction? Protozoa, okay, before I talk about means of reproduction, in terms of nucleus, some members of protozoa are uninucleate. What does it mean to be uninucleate? They have one nucleus. Some are binucleate. Some are multinucleate. Example of uninucleate protozoa. Amoeba has one nucleus. Is a uninucleate protozoa. Therefore, binucleate, paramecium has two nuclei, the larger one and the smaller one. That's the micro and the mega nuclei. Then multinucleate, we have the one they call opalina. So members of opalina are multinucleate. They have more than one nucleus. They have more than one nuclei, sorry. That is for opalina. So these are some of the characteristics of a protozoa. Then in terms of reproduction, In terms of reproduction, I've explained that gaseous exchange occurs within their cell membrane. And it is also important that the presence of water is the singular most important ecological factor that affects the distribution. That means that these parasites, sorry, this organism, you know, are mostly, or for some of them, are mostly distributed within aquatic environment. And when I say aquatic environment, I mean the freshwater environment. So in terms of reproduction, they can reproduce sexually through syngamy, then asexually they can reproduce through binary fission, binary fission, multiple, multiple fission. Then some can also reproduce through budding. The kind of budding is internal budding, which is called endodiogeny. Then some can also reproduce. Okay, okay, that's it. Binary fission, multiple fission, and budding. When it comes to syngamy, of course, that's the fusion of two gametes. This can you can see this in the case of Plasmodium. The life cycle of Plasmodium, of course, you know, the life cycle of Plasmodium has several different stages. You have the schizogony, the pre-exo erythrocytic schizogony, the one that occurs, you know, outside the red blood cell. Then erythrocytic schizogony. Then, of course, we now have syngamy, sexual phase. Then, of course, you now have the sporogony. So you can see syngamy there. Syngamy. There is a union or fusion of the two gametes. That is a form of sexual reproduction that occurs in protozoa. Then we have binary fission, and I've explained binary fission before when we talked about reproduction in protozoa, where uh, where an organism, the parent organism, divides into two daughter cells. First of all, the nucleus divides, and once division of the nucleus is called Carrier, karyokinesis. Carrier. Most words that begin with carrier refer to nucleus in biology. So division of the nucleus is karyokinesis. Kinesis division, and this is followed by the division of the cytoplasm, which is called cytokinesis. So this is what happens, and it separates the cells into two. But for budding, okay, for multiple fission, the nucleus divides into multiple parts. Instead of dividing into two, the nucleus will now divide into multiple parts. And the number of nuclei will determine the number of cells that will be produced from it. So assuming we'll have this 1, 2, 3, 4, 5, 6. Assuming we'll have 6 nuclei produced, that means that 6 different cells will be produced from it. So this is multiple fission. Multiple fission is the same thing as schizogony, and the same thing as sporogony. But what is the difference between schizogony and sporogony? The difference is that schizogony occurs before sexual reproduction, whereas sporogony occurs after sexual reproduction. That is the difference. Schizogony occurs before sexual reproduction, whereas sporogony occurs after sexual reproduction. Just like I explained in the life cycle of a Plasmodium. It begins with schizogony. Then we have the sexual phase before sporogony. And of course, you know that sporogony occurs when the sporozoids begin to divide within the oocysts. Okay, when in the body of the mosquito, when the micro and the mega gametocyte meet together, they form a zygote. This zygote transforms into oconate, and the oconate penetrates the stomach wall of the mosquito, then forms an oocyst. So within the oocyst, the sporozoids now divide into multiple parts before they break and are now released into the proboscis. So that division that occurs after sexual reproduction is called sporogony within the oocyst. So that's the difference between schizogony and sporogony, but both of them are also called multiple fission because in this case, the parent organism divides into multiple parts. In body, we have internal body that occurs in protozoa. It's called endodiogeny. So in this case, the bud grows within the body of the parent, and once it survives, the parent organism is destabilized, so that the bud begin to live an independent life. So that is endodiogeny, and you can see this in Toxoplasma gondii. This is a protozoa parasite that undergoes budding. Toxoplasma gondii. Toxoplasma. Toxoplasma. Toxoplasma gondii. All right. So I think the next thing to talk about now is the fact that protozoa, some are autotrophic, while some are heterotrophic. Some are autotrophic, some are autotrophic, autotrophic, while some are heterotrophic. So what does it mean to be autotrophic? That means the ability to produce your own food. So some protozoa that are found under the phytoflagellates are able to produce their own food, like the chlamydomonas, even the eugleina. They're able to produce their own food. Then the heterotrophic ones are the animal-like. These ones lack the ability to produce their own food. For instance, the trichomonas, the amoeba, and the rest of them, they are all heterotrophic. They cannot produce their own food. So I will end this class by talking about the classification of protozoa. A minute. Classification of protozoa. So protozoa can be classified into four different. Okay, we have the Sarcomastigophora, which is made up of Mastigophora and Sarcodina. We have the Epicomplesia, then have the Ciliophora. We have Microspora. We have Myzosa. So the last three are not usually important. So the last two, are not usually the Microspora and the Myzosa are not usually too important. The most important ones are these first three, Sarcomastigophora. As you can see, this is a combination of Sarcodina, and Mastigophora. Whenever you hear Mastigophora, it's talking about flagellates. So another name for Mastigophora is flagellata. That means that these are the group of protozoa that use flagella for the purpose of locomotion. And I've mentioned an example of Trypanosoma and Leishmania under Mastigophora. Then Sarcodina. Sarcodina is also means amoeba. Another name for Sarcodina is amoeba. So we have the parasitic one, the Entamoeba zoetica. Then the free living one is Amoeba proteus. Then epicomplesia. When we talk about epicomplesia, we mean that this group of protozoa have an apical complex. They have a complex head. They have a complex head, and it's usually found in Plasmodium. And not even in all stages of Plasmodium. Those invasive stages of Plasmodium, I mentioned that when we talked about the life cycle of Plasmodium, like the schizons, and the rest of them, the invasive stages, of Plasmodium, they are equipped with this apical complex. Even the okinet is another one. The okinet is equipped with apical complex so that it can perform its function of invasion at a particular point in the life cycle of the parasite. So example is Plasmodium, then Styroforia is where we see those that use cilia for their locomotion. So this is how far we can go. It is also important you know that members of Epicomplexia, okay, Sporozoa, that's a class. You know, let me also say this. This is a protozoa can be classified this way, but there's another way. There's another way to classify protozoa. It's simpler method. You can use, you can say flagellata, sporozoa, then sarcodina, and ciliata. So some people can also simplify the classification of protozoa this way. Flagellata, flagellata. So this class is where you see that Epicomplexia, which is a subphylum. Epicomplexia, it is okay. Let me say this is under Epicomplexia, or how do I want to put it now? Epicomplexia is also referring to this Sporozoa. Sporozoa is a class of Plasmodium. Epicomplexia is a subphylum. So if we're using this class of classification, that means you're going to replace Epicomplexia with Sporozoa. Then that second Masigofera, we divided it into two. Remember I told you that Masigofera is also called flagellata. So this is flagellata, this is sarcodina. So these two have replaced the Masigofera. Then the Pseudophora was replaced by Ciliata. Okay, so I think this is the only thing we need to study on that protozoa. Of course, you know the function of pseudopodia. Pseudopodia is pseudopodia is used by amoeba for the purpose of feeding. So if you have this as amoeba, then it has a food it wants to get, then it ascends in cytoplasm. Once it ascends in cytoplasm, the cytoplasm helps to engulf the food, and the food is digested within the vacuole. So that is how they feed using the pseudopodia for feeding, mm, and also for the purpose of locomotion because it's a force feed. Why is it a force feed? Because it's not fixed. Any part of the cytoplasm could be extended. That is why pseudopodia is called temporary extension of the cytoplasm. And the process by which protozoa does this, use the pseudopodia to engulf the food particle, is what we call phagocytosis. Phagocytosis. And this is also one of the methods that white blood cells use to engulf pathogens within the body. It is also called phagocytosis, and they use similar method to engulf pathogen within the body. So, ladies and gentlemen, this is how far we can go for today. Thank you for being part of this class. See you in my next class.